Hello, my name is Joshua Mugabi. I welcome you to Biology Remedial Lessons. Today, we are on Remedial Lesson 9, and we are going to discuss about active transport. You are welcome. What is active transport? What is active transport? We can say that active transport is the movement of molecules and ions against a concentration gradient. We are saying active transport is the movement of molecules and ions against a concentration gradient. What we are saying is that when you look at our diagram, it is a diagram showing the cell membrane. There is the exterior that is outside the cell and cytoplasm on the other side that is inside the cell. So it is showing us the molecules, the polar molecules or ions on the exterior. We can see there are two polar molecules. And when you go to the cytoplasm, you can see that we have 7 molecules or ions in the cytoplasm. So it means that the exterior has got fewer polar molecules compared to the cytoplasm. And we are moving these molecules from the exterior to the cytoplasm. So which means that the higher concentration in the cytoplasm of the polar molecules makes the cytoplasm to be more concentrated compared to the exterior. So here outside the exterior, the concentration is low. And when you go to the cytoplasm, the concentration is high. So to move molecules from a region of lower concentration to higher concentration, that one we shall need to use energy to move these molecules or ions against the concentration gradient. So that one is what we are referring to as active transport. We can still say that active transport, the substances move from a lower to a higher concentration gradient by use of energy. We can say active transport, the substances move from a lower concentration to a higher concentration gradient by use of energy. As we have seen across our cell membrane, the exterior has got a lower concentration of polar molecules or ions, and the cytoplasm inside the cell has got a higher concentration gradient. So to move the polar molecules from the exterior into the cytoplasm, that is, we are moving the molecules from a lower concentration to a higher concentration gradient, and that one is going to require us to use energy. So our transporter will need energy to transport the molecules from lower concentration to a higher concentration, and that is what we call active transport. State the factors affecting active transport. State the factors affecting active transport. One of the factors is oxygen concentration. Oxygen concentration, how does it affect active transport? We know that active transport uses energy, and that energy is got from oxidation of glucose in the presence of oxygen to give us carbon dioxide, water, and energy, which we are going to use in active transport. So oxygen is very important because oxygen is the one which is going to oxidize glucose. If we have enough oxygen, then we shall oxidize more glucose and we shall produce more energy. But if we have less oxygen, therefore the amount of glucose which is going to be oxidized is going to be little, and the amount of energy we are going to produce will also be little. That means that we shall be affecting active transport because it requires the use of energy. Another factor is temperature. So how does temperature affect active transport? When we go back to our respiration reaction, this reaction is controlled by enzymes. Enzymes speed up the reaction to provide us with energy. So how does temperature come in to affect the rate of the reaction? Let us try to see. Now, when we look at the temperature, we shall look at our curve. Now, this curve, on the horizontal, we have temperature. On the vertical, we have the rate of reaction of our reaction. And we know that our reaction is controlled by enzymes. So the enzymes are the ones that are going to be affected by the temperature. You can see that when the temperatures are low at around 0 degrees Celsius, the enzyme activities are also low. So the rate of reaction is still also 0. But let us suppose that we increase the temperature from 0 to 20 degrees Celsius. If you try to extrapolate, you will see that as the temperature increases from 0 to 20 degrees Celsius, the rate of reaction also increases. Meaning that when the temperature was 0, the enzyme activity were low and the rate of reaction was low. The enzymes were inactive. But when we increased the temperature from 0 to 20 degrees Celsius, we saw that also the enzyme activity increased. And when they increased, the rate of reaction increased and the products that we produced also increased. Now let us say we increase the temperature furthermore from 20 and we take it to 40. So when we extrapolate at 40 degrees on our curve, we can also see that the rate of reaction increased, meaning that the enzyme activities also increased. So this means that as we increase the temperature, we also increase the enzyme activity. So we look at the 40 degrees Celsius, the enzymes are more active. What if we increase the temperature further? What would you expect? Do you expect the enzyme activity to increase or to reduce? Because we are seeing the trend is that as we increase the temperature, the enzyme activity increases and the reaction rate also increases, meaning that our energy, we are getting more energy. Let's say we increase the temperature beyond 40 and we make it around 50 degrees Celsius. Now, when we extrapolate, we can see that as we increase the temperature from 40 to 50, again now, the reaction rate is reducing. What is happening? What is happening here is that as we increase the temperature from 40 to 50 degrees Celsius, the rate of reaction is decreasing. What is happening is that now the higher temperature is denaturing the enzyme. And when you denature the enzyme, it means that the enzymes now cannot perform their work. and therefore the rate of reaction is going to reduce. So that means that the optimum temperature where the enzymes work best is at 40 degrees Celsius. That's when the enzyme will perform to their best. So we can conclude by saying that one of the factors affecting enzyme activities is temperature, because it is going to affect the enzyme activities, and when it affects enzyme activity, it is going to determine the amount of energy we are going to produce, and this energy is the one which is going to determine the rate of active transport. Another factor is the change in pH. How does the change in pH affect active transport? From our reaction, respiration reaction, we know that this reaction is controlled by enzyme. So how does pH come in? Let's see how pH affects enzyme activity. Now, we have the pH curve, and we have the enzyme activities. So on the horizontal, we have the change in pH. Vertical, we have the rate of reaction. Now, this enzyme works best at a pH of 7, which is its optimum pH, meaning at that pH, this enzyme is very active. When the enzyme is provided a pH of 5 and you extrapolate, you will see that its rate of reaction is low. The rate of reaction is low, meaning that the enzyme does not work well in acidic medium. What if we take the pH and give it a 7 pH, which is a neutral pH? When we provide it with a neutral pH, which is around 7, and we extrapolate on our curve, we also see that the rate of reaction increases. This means that this enzyme works best at a neutral pH. We know that neutral pH is 7. So it works best at a neutral pH, because we are seeing that when we provide a pH of 7, the rate of reaction increases. What if we change the pH and take it around 8? 8, that one now is becoming more alkaline. And when we extrapolate, we can see on our graph that when we provide a pH of 8, the rate of reaction reduces. This means that the enzymes do not work well in alkaline medium. So by changing the pH from neutral to alkaline, that one is going to affect the activity of the enzymes, because as you make the pH alkaline, you are denaturing the enzyme. You are changing the conformity, or you are distorting the structure of the enzyme. And as you distort the structure of the enzyme, it means that the active site is going to be distorted. Therefore, the enzyme will not be able to fit in the substrate, and its activity will be affected. Therefore, we say that the optimum pH for this enzyme is at 7, which is the neutral. So when you change the pH from any of the either side, either you make it acidic or you make it alkaline, then you are going to affect the enzyme activity. So we can conclude and say that change in pH is going to affect the enzyme activity. And when it affects the enzyme activity, it is going to affect the amount of energy we produce, which will also affect the activity of active transport. Another factor is glucose concentration. How does glucose concentration affect active transport? We know that glucose is our oxidized product, our substrate, which we are oxidizing using oxygen to give us carbon dioxide, water, and energy. So if we don't have enough glucose, that means that the products that we are going to produce, which is energy, is going to be little. But if we have a high concentration of glucose, provided that the other factors are constant, then we shall produce more energy. So we can say that glucose concentration will affect active transport. Another factor are the enzyme inhibitors. Enzyme inhibitors. Knowing that the respiration reaction involves the breakdown of glucose in the presence of oxygen to give us carbon dioxide, water, and energy, this reaction takes place in the presence of enzymes. And enzymes can be affected by inhibitors. And when the inhibitors affect the enzyme, therefore, we are going to affect active transport. How do enzyme inhibitors affect active transport? Let us see. We shall start with competitive inhibition. In competitive inhibition, we have our enzyme. And this enzyme has got an active site where the substrate fits precisely. And we also have our substrate that fits precisely into the enzyme active site. But again, we also have the competitive inhibitor. This competitive inhibitor has a similar shape to the substrate and fits into the enzyme's active site. So this means that these two, the substrate and the competitive inhibitor, they are competing for the active site. If in the situation where we are having the enzyme working on the substrate and in the environment, they are also competitive inhibitors, this one is going to affect the rate of reaction. If we have more substrate concentration compared to the competitive inhibitor, the rate of reaction will not be affected much. But if the competitive inhibitors are more compared to the substrate, then the chances of the substrate colliding into the enzyme active site is going to be reduced. So that one means that the competitive inhibitors will be the one occupying the active site all the time. So the rate of reaction is going to be affected, and the products that we are going to produce are going to be low. That is what we have when we have competitive inhibition. The competitive inhibitor fighting for the active site of the enzyme with the substrate. Then another one is where we have the non-competitive inhibition. In a non-competitive inhibition, we have our enzyme with its active site. And then we have a non-competitive inhibitor. Non-competitive inhibitors, these are other molecules which may bind elsewhere on the enzyme, but not at the active site. They just bind somewhere else on the enzyme, But when they do that, they distort the active site. And when the active site has been distorted or the shape has been changed, that means that the substrate now cannot fit properly into the active site. And if the substrate cannot fit into the active site, that means that we are affecting the activity of the enzyme. The enzyme cannot work properly. Therefore, the non-competitive inhibitors, by binding somewhere else on the enzyme, not competing for the active site with the substrate, what they do, they distort the active site of the enzyme, and the enzyme cannot perform its work. Therefore, we can conclude and say enzyme inhibitors affect active transport, because when the enzymes are affected, they cannot work well, and therefore the rate of reaction will be low, and the amount of energy we are going to produce is going to be little, which is going to affect active transport. Why is oxygen important in the process of active transport? Why is oxygen important in the process of active transport? Now, oxygen is required for respiration, which produces energy necessary for process to occur. So we know that for us to get energy, we need to oxidize glucose to give us energy. And this reaction, the respiration reaction, requires oxygen. So if oxygen is not provided, then we are not going to produce our energy. So oxygen is required for respiration reaction to take place, so that we are able to produce energy, which is necessary for the active transport process. Another factor affecting the rate of active transport in living organism is oxygen concentration. It is required for respiration or to oxidize respiratory substrate to release energy required for active transport. Oxygen is required for respiration, or oxygen is required to oxidize the respiratory substrate, and our respiratory substrate is oxygen, is a glucose. And when it is oxidized, it is going to release energy, which is required for active transport. So oxygen is a factor which will affect active transport, because it is the one which oxidizes our respiratory substrate, which is glucose, to release energy, which is required for active transport. An increase in oxygen concentration causes a simultaneous increase to the rate of active transport up to a certain level. So when we increase the oxygen concentration, at the same time we are also increasing the rate of active transport up to a certain level. Another factor affecting the rate of active transport in living organism is pH. We know that enzymes, being protein in nature, are specific. Enzymes being protein in nature, they are specific. And we know that enzymes, they work in a specific pH. Most of the enzymes, they work in a neutral pH, which is around 7. That is their optimum pH. But if we change the pH to become acidic, that is below 7, then we are going to denature the enzymes. They will not work well. The rate of reaction will reduce, and thus we shall be affecting active transport because we shall be producing less amount of energy. And if we also make the pH to become more alkaline, that is beyond 7, that one also is going to reduce the enzyme activity, and the rate of reaction will be low, and we shall produce less amount of energy. Therefore, we shall be affecting the rate of active transport in living organisms. So we can say that the extreme change in pH affects the rate of respiration, which is controlled by enzymes, and may denature the enzymes, reducing the rate of active transport. The extreme change of the pH, if you extremely change the pH, make it alkaline, make it acidic, it is going to affect the rate of respiration. And we know that the respiration reaction is controlled by enzymes. And if we change the pH extremely, then we are going to denature the enzymes. And when you denature the enzyme activity is going to reduce, and thereby we are going to affect the rate of active transport. Another one is glucose concentration. Glucose is the main respiratory substrate for energy production. It is the main respiratory substrate for energy production. So when we have our glucose enough and we oxidize it, we shall get enough energy, and therefore the rate of active transport will be faster. But if we have less glucose concentration, then the amount of energy we are going to get is going to be less. And if you have less amount of energy, then the rate of active transport in living organism is going to be affected. So, an increase in glucose concentration in cells increases the rate of respiration, and hence the rate of active transport is increased up to a certain level. So when we increase the glucose concentration in the cell, at the same time we are increasing the rate of respiration. And when we increase the rate of respiration, that means that we are producing more energy. And this more energy is going to cause the increase in the rate of active transport up to a certain optimum level. So glucose can affect the rate of active transport in living organisms. Another factor is temperature. The process of respiration by which energy for active transport is generated is controlled by enzymes. We know that the process of respiration, that reaction of respiration where we oxidize glucose to give us energy, is controlled by enzymes. So being that it is controlled by enzymes, temperature is going to affect the enzyme activity. If the temperatures are around 37 to 40 degrees Celsius, that is the optimum temperature for enzyme activities. The enzymes will perform to their best at 40 degrees Celsius, which is the optimum temperature. But if we reduce the temperatures, that is the temperatures are below 37 to 0, the enzymes are inactive, meaning that the rate at which they are working is very slow. So the rate of reaction will be low. But if again we increase the temperature and it goes beyond the optimum temperature, 40 and above, then the enzyme's rate of reaction is going to reduce, meaning that the enzymes are being denatured. So they are going to be affected. So we can say enzymes work best at a temperature of between 35 degrees Celsius to 40 degrees Celsius, usually called the optimum range. So the temperature at which enzymes work best is between 35 degrees Celsius and 40 degrees Celsius. And this one is what we call the optimum temperature. So temperature can affect the rate of active transport by affecting the activities of the enzymes. At very low temperatures, enzymes are inactive, lowering the rate of respiration, hence low rates of active transport. So when the temperatures are low, we are going to make the enzymes inactive if the temperatures are very low. And that one means that the rate of respiration is also going to be very low in turn, which is going to reduce the rate of active transport. So increasing in temperature above optimum temperature, which is above 40 degrees Celsius, denatures enzymes, slowing down respiration and active transport until it finally stops. When we increase the temperature above the optimum temperature, which is 40 degrees Celsius, this one is going to denature the enzymes. And when the enzymes are denatured, it means that we are slowing down respiration. And at the same time, that one will slow down the active transport until when time comes, it finally stops. So temperature is affecting the rate of active transport in living organisms. Another factor are enzyme inhibitors. Now, enzyme inhibitors, they are substances which slow down or stop the activity or functioning of the enzymes. These are substances that are going to slow down, or they can also stop the activity and the functioning of enzymes. How do they do it? They do it by competing with the substrate for the active site in the enzyme, or they can block the active site of the enzyme. So when they do that, they are inhibiting the substrate to access the active site of the enzyme. So they will inhibit the activity, the activity and functioning of the enzyme. And by doing so, we shall not be able to get enough energy, and hence we shall be affecting the rate of active transport in living organisms. Another one is the concentration of carrier molecules in the cell membrane. Concentration of carrier molecules in the cell membrane. Carrier molecules, they are substances that bind to the ions being transported actively and carrying them across the membrane. The carrier molecules, they are substances that bind to the ions that are being transported actively, meaning that we are using energy across the membrane. Those are what we call carrier molecules. They are substances that help to bind the ions that are going to be transported across the membrane, but this time they are transporting them using energy. Those are carrier molecules. Increasing the concentration of carrier molecules, increasing the rate of active transport up to a certain level and vice versa. So we are seeing that when we have very many carrier molecules or a high concentration of carrier molecules on the membrane, this one is going to increase the rate of active transport. Because the more the carrier molecules you have, the more substrates or ions or molecules will be transported. And when you do that one, that means that you are increasing the rate of active transport. And if their number is lower, the number or the concentration of carrier molecules is lower, that one will also reduce the rate of active transport because less molecules are going to be carried across the membrane. So the concentration of carrier molecules in the cell membrane affects the rate of active transport in living organisms. Explain briefly the role of active transport in living organisms. Explain briefly the role of active transport in living organisms. Active transport is involved in active reabsorption of glucose and amino salts in the kidney tubuli during the formation of urine. We are saying active transport is involved in the active reabsorption of glucose. Active reabsorption, meaning that we are using energy to reabsorb glucose and amino salts in the kidney tubuli during formation of urine. So we use active transport to reabsorb glucose and amino salts from the kidney tubuli back into the blood vessels. So when you look at our nephron structure, we have that part which is called the proximal convoluted tubuli. In the proximal convoluted tubuli, that is where glucose and water are reabsorbed back into the blood capillary. And it can be by diffusion or by active transport. And then when you go below the proximal convoluted tubuli, that one now you are going towards the descending limb of Henle. There, salts and more water are absorbed back into the blood capillary. And those ones also require use of energy. So we say that active transport in living organisms is involved in the reabsorption of glucose and amino salts in the kidney tubuli during the formation of urine. Now, it also enables the absorption of digested food from the alimentary canal or the small intestine into the blood stream. So, active transport is going to be used in the absorption of digested foods from the alimentary canal or the small intestines into the bloodstream. So when we look at our diagram here, some of the food substances that have been digested, we have the amino acids, we have the glucose, and then we have the fatty acids and glycerol. So some of these substances can be absorbed against the concentration gradient. The amino acids will be absorbed into the blood capillary from the alimentary canal, and the glucose will also be absorbed into the blood capillary from the small intestine. Then we have the fatty acids. These ones are going to be absorbed into the lactate, still from the small intestine. So sometimes they are absorbed against the concentration gradient. That is where they are going. There is a high concentration, and in the alimentary canal, their concentration is low. So there we shall use active transport to absorb them from the small intestine into the capillaries and the lactate. Active transport still in excretion of waste product from the body cell for eventual removal. It is used in excretion of waste products from the body cells for eventual removal. Now, we have our cell here. It recognizes the bacteria. Then it engulfs the bacteria. It forms a phagocytic vacuole. This phagocytic vacuole is encountered by the lysosome containing the digestive enzyme, which fuses with the phagocytic vacuole, and the bacteria is digested. And then afterwards, the undigested materials are removed by exocytosis from the cell. So that is removing those undigested materials from the cells, which are eventually removed from the body. That activity of engulfing and discharging the undigested material requires energy, and we use active transport. Then active transport is also involved in the transmission of nerve impulses in the nerve cells through sodium pump, and maintains a balance between sodium and potassium ions. It is involved in the transmission of nerve impulses. Where are they taking place? In the nerve cell. So we are going to use the sodium-potassium pump. And when you hear that word pump, it means that we are going to use energy. So active transport is going to be involved in the transmission of nerve impulses within the nerve cell through the sodium-potassium pump. And this one is the one that helps us to balance between sodium and potassium ions in and outside the cell. Now, when you look briefly at our cell, a cross-section of the nerve cell, you will see inside the cell where we have the axon cytoplasm, we have low concentration of sodium and high concentration of potassium. But when we come to the surrounding of the nerve cell, what we are calling the tissue fluid, it has a high concentration of sodium and low concentration of potassium. Now, that means that the low concentration in the axon cytoplasm and the high concentration in the tissue fluid of sodium ions makes the inside of the axon or the nerve cell to become negative because it has got low concentration of sodium ions compared to the tissue fluid. So what happens is that when the nerve impulse is coming and is going to pass that section of the nerve cell inside the axon cytoplasm, it has to be positively charged. The nerve impulse cannot pass when inside is negatively charged. So how can we make inside the positive so that the nerve can pass? Now, we have these structures down here. These are called voltage-gated channels. They are voltage-gated channels. These ones, they can open and allow sodium to flow from the tissue fluid to enter into the oxygen cytoplasm. By doing so, we shall increase on the concentration of sodium in the oxygen cytoplasm, and inside it will become positive. So the channel gated voltage gated channels will open, and when they open, sodium ions are going to diffuse from the tissue fluid and they will enter into the oxygen cytoplasm. And then the potassium ions inside the oxygen cytoplasm will diffuse and go into the tissue fluid. They will diffuse and go out. So by increasing the concentration of sodium inside, that means inside becomes more positive and the negativity goes away. And when it becomes positive, that means now our nerve impulse can pass. And when the nerve impulse has passed, that means that we no longer need the work of the voltage gated channels. They will close. So the voltage gated channel will close, and when it closes, that means no more entry of sodium ions into the oxygen cytoplasm and no more allowing potassium to get out of the oxygen going into the tissue fluid. But now the nerve impulse has passed, and we need now to take back the charge inside the oxygen cytoplasm back to negative. How can we do that? That one is going to be done by reducing the concentration of sodium in the oxygen cytoplasm by taking it out so that inside becomes negative. How are we going to do that? Now, we shall use energy, which is stored in form of ATP. ATP is going to break down into ADP plus phosphate ions. And when it breaks down, it is going to provide us with energy. Now, this energy is going to help us to use the sodium-potassium pump. Will start working. And as it is working, it will push out the sodium ions from the oxygen cytoplasm into the tissue fluid. And by doing so, it also pumps in potassium ions from the tissue fluid into the oxygen cytoplasm. And by doing so, the number of sodium ions inside the oxygen cytoplasm is going to reduce. And as it is reducing, the positivity is going to change into negativity. And then our oxygen cytoplasm will go back to its original potential, which is negative potential. And therefore, now we no longer need the use of energy. We shall stop using the energy. And then when we stop providing the energy, then our sodium-potassium pump will also stop, because the oxygen cytoplasm has gone back to its negative potential. So that is how active transport is utilized in living organism, especially being involved in the transmission of nerve impulses within the nerve cell through the sodium-potassium pump to maintain a balance between sodium and potassium ions. Now, how is it involved in the transportation of mineral salts and water? In plants, it enables plant roots to absorb water from the soil against the concentration gradient. So we are transporting water against the concentration gradient. But when we look at our diagram here, if we look at our diagram here, it is saying dilute solution soil, meaning that the soil has got a higher water potential. And when you look at the concentration in the vacuole and the cytoplasm of the root hair, there is high concentration, meaning that there is low water potential. That means water is going to move by osmosis. It will diffuse down the water potential gradient from the soil, which has got a high water potential, into the virtue and the cytoplasm of the root hair, which has got a lower water potential. But it is not always like that. We have some plants that are found in arid areas or dry areas. Their soil does not have a high water potential. It has a lower water potential. And sometimes you may find the root hair in those plants in arid areas have got a higher water potential, but they still need to absorb any amount of water in the soil. So that means that the water which is in the soil is going to be transported against the concentration gradient, because in the cytoplasm and the virtue of the root hair of arid plants, they have more water, meaning that they have a high water potential, and the soil surrounding the root hair has less amount of water, so meaning it has lower water potential. So there is no way water will diffuse from the soil into the root hair by osmosis, because the root hair has got a higher water potential compared to the soil. So if we need to get this water from the soil into and take it into the root hair, virtue and cytoplasm, we have to use energy, and there active transport comes in. So water will be absorbed by the root against the concentration gradient, and there we shall use energy, which is active transport. Now, when it comes to transport of manufactured food substances, active transport will be involved in the translocation of manufactured food in the phloem tissue within the plant body. It is involved in the translocation of manufactured food in the phloem tissue within the body of the plant. How is it done? Now, we have our phloem sheath element. Next to it is the companion cell, and far away from it is the source of our manufactured food, or the leaf which is carrying out photosynthesis. So how is active transport involved in the translocation of the manufactured food through the phloem tissue? Now, here, when you look at the companion cell, in the companion cell, we have a lot of hydrogen ions. Now, these hydrogen ions, by the use of ATP energy, we are going to push them from the cytoplasm of the companion cell and push them into the cell wall of the companion cell. So when we push those hydrogen ions into the cell wall, their concentration is going to increase in the cell wall. And as they increase in the cell wall, then in our source, or the leaf where photosynthesis is taking place, it is now going to attract or absorb glucose. And glucose will enter into the cell wall, and when the hydrogen ions in the cell wall are being transported back into the companion cell cytoplasm, they will go with glucose. So there is going to be a co-transport. That co-transporter, which is transporting hydrogen ions from the cell wall back into the cytoplasm of the companion cell, will transport the hydrogen ions at the same time transporting the sucrose. So that means that now, as the hydrogen ions are coming from the cytoplasm, entering back into the cytoplasm of the companion cell, the transporter carrying the hydrogen ions is also transporting sucrose, which is coming from the source or the leaf cell where photosynthesis is taking place. And very soon, we shall have a higher concentration of sucrose in the companion cell cytoplasm. When that one happens, now the sucrose will cross from the companion cell to the phloem sieve element via the plasmodesmata. Plasmodesmata is a connection between the companion cell and the phloem sieve tube. So it will flow along the concentration gradient. Because there is a high concentration of glucose in the companion cell and low concentration in the phloem sieve tubes, so it will flow from a region of high concentration to a region of low concentration. And the region of high concentration of sucrose is now the companion cell, a region of low concentration at the phloem sieve tube. So sucrose will flow from the companion cell into the phloem sieve tube. Now, when it enters into the phloem sieve tube, its concentration is also going to increase. And as its concentration is increasing, it is going to absorb water from the neighboring cells or tissue. The water will enter into the phloem sieve elements by osmosis, being attracted by the sucrose concentration. So moving from the region of low water concentration to a region of moving from a region of high water concentration, those are the nearby cells, into a region of low water concentration, that is the phloem sieve elements where there is sucrose. And when that one happens, water moving from neighboring cells entering into the phloem sieve, then we are going to have a mass of glucose mixed with sucrose mixed with water, and then they will start flowing through the sieve pores by mass flow. So you can see that the active transport has been involved in the translocation of manufactured food in the phloem tissue within the plant, because it started by using energy to transport the hydrogen ions from the companion cell to the cell wall, which initiated the whole system of translocation of sucrose from the source into the phloem sieve tube. Now, it is also involved, active transport is also involved in the opening and closing of the stomata through the sodium-potassium pump mechanism. Active transport is involved in the opening and the closing of what? Of the stomata. Through what? Using the potassium-sodium pump. And whenever we are talking of a pump, it means we are using energy. So let's see how it takes place. In the gut cell, we have hydrogen ions, and we have a hydrogen pump. Now, that hydrogen pump uses energy, which comes from ATP. It breaks down ATP to ADP plus phosphate ions and provides the pump with energy. And when the pump is provided with energy, it is going to pump the hydrogen ions out of the gut cell to the surrounding cells. And when that one happens, it means that now the concentration of hydrogen ions in the gut cell is going to reduce. And when it reduces, inside the gut cell becomes negatively charged. And because of the negative charge inside, the potassium ions, which are outside the gut cells, are going to be attracted to enter way into the gut cell where it is negatively charged. That is electrical gradient. And they will also enter because of the concentration gradient. Potassium ions are high concentration outside the gut cell, and they are on low concentration inside the gut cell. So because the potassium channels have opened, potassium will diffuse from the nearby cells into the gut cell. And when potassium keeps on diffusing into the gut cell, that one means inside it becomes more positive. And we have more concentration of potassium ions inside. When the potassium concentration increases into the gut cell, that one is going to reduce the water potential inside the gut cell. And when we have low water potential in the gut cell, now that one is going to cause water. It is going to cause the water to move from a region of high water potential into the gut cell which has got low water potential. So water will flow from the nearby cells entering into the gut cell. And that one, as it continues happening, the gut cell is going to start enlarging and swelling. And as it swells, the gut cell will become tagged. And when it becomes tagged, the stomach will open. So we are seeing that active transport is involved in the opening and closing of the stomata through the sodium-potassium pump mechanism, which requires energy. So active transport is active in the opening and closing of the stomata. Outline the roles of active transport in a living organism. Now, it is used in mineral salts intake by plant. We have talked about that one. There is a dilute high water potential in the soil, low water potential in the root, and then water will move by osmosis, but we say that in most cases, soil is going to have a lot of less minerals than compared to those ones in the plant. So the minerals will be absorbed by active transport. Selective reabsorption of glucose and some salts in the kidney. So we have looked at the reabsorption of water and glucose in the distal or proximal vaulted tubule, and the absorption of salts and water from the renal lobe of Henle back into the blood capillary. Still there we are using energy, which is active transport. Then we have looked at the absorption of digested food by the small intestine, where we say that digested food is absorbed into the blood capillaries. We have the amino acids have been digested, glucose has been digested, we have the fatty acids and glycerol, and we have said that the amino acid will be absorbed into the red blood capillaries, and glucose will be absorbed into the red blood capillary, fatty acids and glycerol will be absorbed into the lacteal, and thereby active transport is being involved in the absorption of digested food in the small intestines. Then we talked also about the excretion of waste products from the cells. We saw that some waste products can be removed from the cell. For example, we looked at the red blood cell, whereby it recognizes the presence of a bacteria. The bacterium is engulfed, and then we end up forming a phagocytic vacuole. This phagocytic vacuole is encountered by lysosomes containing digestive enzymes, which fuses with the phagocytic vacuole, and then the bacterium is digested, and after digested, of course, their undigested remains of the bacterium can be now removed by exocytosis. And exocytosis and the engulfment of phagocytosis, all those processes, they require energy. So active transport is involved in the excretion of waste products from the body cells. Then we talked about the sodium pump mechanism in the nerve cells or the neuron. And we say that inside the oxygen cytoplasm, low concentration of sodium, high concentration of potassium. And then in the tissue fluid, we have high concentration of sodium, low concentration of potassium. And that makes the inside of the neuron to become negatively charged. So we have the voltage-gated channels. Those ones will open when the nerve impulse comes, so that sodium ions flow inside the oxygen, and then potassium ions flow out of the oxygen. When that one happens, inside the oxygen is going to become positively charged, and now our neuron nerve impulse can pass. And when it has finished passing, we need to remove all the channels. The voltage-gated channels should close, so that no more sodium is allowed to enter inside and no more potassium is allowed to get out of the oxygen. But now the impulse has already passed. We need to take back the potential back to its original potential. So what we are going to do, we are going to use energy. That energy will provide energy to the sodium potassium pump, which is going to pump the sodium outside of the oxygen and pump potassium inside the oxygen. And by doing so, we are going to change the positivity inside back to the negative potential inside the neuron. And when inside it has gone back to its original potential or the resting potential, now we no longer need to use energy. We shall cut off the supply of energy, and then the pump will start pumping sodium out and then pumping potassium inside. It will stop, and then that is whereby we see that the inside is maintained at a negative potential. So we have seen that also active transport also is involved in sodium pump mechanism in the nerve cell or the neurons. Hello, my friends. Thank you very much for participating in remedial lesson 9, where we have talked about active transport. Let's meet again in remedial lesson 10, where we shall discuss about nutrition. Thank you. Please do not forget to press that subscription button. Bye-bye.